Hello, this is the next video that I'm going to do on the nervous system. So this is on the eye. So booklet 4 for the pre-med and pre-dental booklets from page number 15. On page number 15 it has the structures of the eye that you need to be familiar with and their functions. Now we're going to have a look at a number of features of the eye, but before you do that I would suggest that you pause the video now and have a go at completing this worksheet, which is available in the Teams area for the pre-med and pre-dental. So it has a number of different labels, labelled A to PQR, and those match up with the descriptors, the descriptions down below. So if you can match those descriptions to the letters, and to do that you may need to use page 15 from your notes. Okay, so I'm not going to go through each of the structures and each of the functions now, but you're welcome to ask me at a later date if you need anything clarifying about that. So what we will do is start on the eye and how the pupil size can be altered and how the lens or the cornea and the lens are able to focus beams of light onto the retina. So firstly, in your booklet on page 16 there is this diagram, unlabelled diagram of the eye. I'll give you those labels in a moment. Using that worksheet that I've mentioned already, I'd like to match up the structures with the role or the function of each structure, and then after that we'll move on to how the pupil size is controlled, explaining why that is important, and then describe how the lens size is controlled, again explaining why that is important. Okay, so like I said, page 16 in your notes you have this diagram which relates to, relates to this labelled diagram. So my advice would be to pause your video now, give yourself time to label the diagram, and then after you've labelled the diagram, the task is to complete the eye structure and function worksheet. Ooh, I've forgotten how to spell. Worksheet. So if you can pause that video now, use the information on page 15 and this diagram to complete that worksheet. Okay, hopefully you've had a chance to do those, made those labels, and you've completed that worksheet, and now we'll move on to how the pupil size, well, the function of the eye. So this diagram, let me just zoom in a little bit, shows how a beam of light will pass through first the cornea. Let me get my pen up. Okay, so through the cornea, through the lens, and be directed onto the retina. Now the retina contains photoreceptor cells called rods and cones, and we'll come back to those later on. What you may notice from this diagram is that the rods and cones are towards the back of the retina. Just behind those rods and cones is another structure called the choroid. That choroid contains lots of cells containing melanin, which absorbs any light that has passed all the way through the retina without stimulating rods and cones, and that prevents reflection. In front of those rods and cones, so if we imagine that the light is travelling in this direction, so in front of those rods and cones we have two more cells or tissues that you need to be familiar with. The bipolar cells and then the sensory neurons. And it is those sensory neurons which will transmit impulses from the retina, from the eye, and to the visual sensory cortex in the brain. So, as we said, to form an image, light is reflected from an object. So that light that has been reflected from the object will enter the eye. It will first pass through the cornea, and the cornea is responsible for most of the refraction, the bending of the light, what we could think of as the focusing. The lens then does any fine adjustments, so makes fine adjustments to ensure that the image is focused directly onto the retina. From there, the rods and cone cells will be stimulated in the retina. They will produce impulses. Those impulses are transmitted firstly across the bipolar neurons and then the sensory neurons to the optic nerve. The optic nerve travels from the eye to the visual cortex at the back of the head, and in the visual cortex it will first reach the visual sensory cortex. So the brain contains this visual cortex. The first part of the brain that is stimulated is the visual sensory cortex, which receives impulses from the optic nerve. It will then transmit impulses from the visual sensory cortex to the visual association area. At the visual association area, those impulses from the visual sensory cortex are interpreted. And by that we mean the images or the impulses are linked to previous experience and recognisable images. We're going to concentrate now, I was going to say focus, on the control of light intensity. So the amount of light which is able to reach the retina. This involves an adjustment of the pupil size, and this adjustment is autonomic, it's a reflex adjustment, and it involves antagonistic muscles, so circular muscles and radial muscles, which together alter the size of the pupil, the aperture of the pupil, and therefore allows controls the amount of light that is able to pass through the pupil and reach the retina. Here we can see two images. In that image at the top, we can see that there is and the one below. So obviously in a human, we can see here that the pupils are dilated. The pupil size is much larger. This would allow more light to enter the eye and reach the retina. In a cat below, cats have vertical pupils. They get wider as they dilate to allow more light to enter. And here we can see a little a GIF of that, I believe. So we can see the adjustment to the pupil size. Now that adjustment is determined by the antagonistic relationship between circular muscles and those circular muscles, this isn't a great circle, surround the pupil in a circular fashion, and radial muscles, and the radial muscles extend from the pupil and outwards. Okay, so we have circular and radial muscles. Those circular and radial muscles, as I've said before, are antagonistic, meaning that as the circular muscles contract, the radial muscles will relax, and vice versa. Okay, this shows the reflex arc between the eye, between those the receptors in the eye will send impulses to the central nervous system. There is a relay in the central nervous system. So the receptors send impulses via a sensory neuron to the central nervous system in the spinal column, or in the central nervous system, really. I think what's it called? Anyway, never mind. And then a motor neuron will transmit those impulses from the central nervous system back to the circular and radial muscles in the eye. So we'll look at this in a bit more detail then. As the photoreceptors are stimulated, impulses or action potentials are generated, and impulses travel along the sensory neuron and where they reach the central nervous system and a relay neuron. The relay neuron will then transmit those impulses to the motor neuron, causing either the radial muscles or the circular muscles to contract and the others to relax. In bright light, to limit the amount of light that enters the eye, or to limit the light intensity, the pupil becomes smaller. So as with all reflex arcs, this is to prevent damage to tissues. So by limiting the amount of light that enters the eye, that prevents perhaps damage to the retina itself, but it would also mean that the retina is able to form an image without being bleached in high light intensity. Now we'll come back to that concept later on. Sorry, let's return to this. Okay, so in bright light, to make the pupil size smaller, the circular muscles contract. So those circular muscles which travel around a pupil contract, causing the pupil to become smaller. At the same time, the radial muscles relax. In dim light, the pupil dilates. Now this is necessary to ensure that enough light is entering the eye to form an image, and again that could be related to reducing tissue damage. So perhaps a pupil will dilate, for example, if there is a threat, a predator, for example, and that means that more information can enter the eye, so an organism is able to respond to that potential threat and therefore prevent damage to tissues. So bright light and pupils contracting, becoming smaller, is parasympathetic, whereas dim light and the pupil dilation is controlled by the sympathetic nervous system. Now these 2 points are important. I think there are 2 questions in your question booklet that relate to this. Okay, so the sympathetic nervous system causes those circular muscles to relax and the radial muscles to contract. As the radial muscles contract, that pulls the pupil, increases the size of the pupil by pulling that iris away from the pupil. The parasympathetic control of pupil size, the circular muscles contract, radial muscles relax, and the pupil becomes smaller. Okay, so here we can see that the pupil is constricted under bright light. That prevents overstimulation of the receptors, which could potentially cause retinal damage, damage to the retina, damage of those tissues that we mentioned earlier on. In dim light, the pupil dilates, more light enters the eye to stimulate the retina, enables a person to see better and perhaps take in more information. As we said earlier on, constriction is controlled by the parasympathetic nervous system, dilation controlled by the sympathetic nervous system. Okay, now these are some terms that I'm going to introduce that aren't in your notes, but you may be interested in. So the first is meiosis. Without the E, this isn't a form of cell division to make gametes. This can be caused by, so we can see here the pupil constriction. So this can be caused by opiates, and it is a fairly, what am I thinking of the word? It's a sign, I guess. There is a better word, but it's failed me at the moment. A sign that a person may be showing heroin intoxication, where there is considerable constriction of the pupil. Opiates as well, with heroin withdrawal, can result in dilation. Nicotine as well has a similar effect, and so do some antipsychotics. Mydriasis is the opposite here, so we can see that the pupil has dilated in this image. Any drug which increases brain serotonin levels, such as MDMA, cocaine, perhaps antidepressants, and the hormone oxytocin, or perhaps physical trauma and head injury, can all be responsible for mydriasis, this dilation of the pupil. Okay, so we'll move on now to accommodation, or focusing, and producing a focused image on the retina. So this is achieved by fine adjustment to the shape of the lens, and that will change the converging power. So it changes the amount of refraction of light to ensure that the image is formed on the retina and not before or after it. I'll explain that a bit further later. So this is achieved by contraction and relaxation of the ciliary muscles. Here we have the components then for accommodation, those parts of the eye that are responsible for this. These larger structures here are known as the ciliary bodies, and the ciliary bodies contain the ciliary muscle. Then we have the suspensory ligaments from the ciliary muscles onto the lens itself. Here we have a dissection of an eye and the removal of the lens. And what you'll notice here is that the lens is really elastic, and in its in its sort of resting state, I suppose, it becomes very spherical. Okay, very circular and spherical. To change the shape of the lens, those ciliary muscles can either relax or contract. Now that will be dependent on the object which is being viewed. A distant object will reflect rays of light which are close to parallel, and therefore will require less refraction. So a distant object with its close to parallel light rays can be refracted by a flattened lens. So a flattened lens is one which is under tension. Okay, as tension is placed upon the lens, it will become more flattened and less circular, less spherical. A close object will produce rays of light, or will reflect rays of light, which we can refer to as diverging, moving away. Now, those diverging rays will require a stronger refraction to produce an image on the retina. For that, the lens must be more spherical. So a more spherical lens will produce greater refraction to take those diverging light rays to converge them onto the retina itself. Okay. In the in this blue box here, it says that some images showing accommodation show light rays meeting at the same point on the retina. This really is to simplify what's happening. It's to make clear that it is producing a focused image on the retina. However, it is slightly misleading. That's not exactly what's happening. In reality, the divergence will cause a focal point, we can see in these diagrams, before the retina. And in fact, an inverted image is produced on the retina, which is later interpreted when those impulses reach the visual association area. But again, we see this same principle. A close object with divergent rays requires greater convergence and therefore a more spherical lens to produce that focal point. A distant object with nearly parallel rays will require less convergence, and therefore the lens is flattened. So we need to understand how the shape of the lens can be altered. With light from a near object, it's necessary to have a lens which is more spherical. As I said, the lens itself is highly elastic, and it will return to that spherical shape unless there is tension applied to it. So when the when the eye is focusing on a near object, the ciliary muscles will become contracted. So to focus on a near object, shown here in both diagrams by number 3, the ciliary muscles, oops, I'm just going to show that, and all the way around, the ciliary muscles contract. They increase in size, which means that the suspensory ligaments become slack. Okay, we can see that in that top image, number 4, the sensory ligaments slackening, and number 4 here, becoming slack as well. So there is less tension on the lens, and the lens, due to its elasticity, will return to a more spherical shape. As it's more spherical, we get greater convergence, focusing those diverging rays of light onto the retina. A distant object with more parallel rays of light require less convergence and therefore require a flattened lens. To achieve that, there needs to be tension on the suspensory ligaments. So with a distant object, the ciliary muscles will relax. The circular ciliary muscles relax. We can see they become smaller, number 3 on both diagrams, compared to number 3 in the previous diagram. Now, as they become smaller, that increases the tension on the suspensory ligament. Taut just means to tighten. Okay, it becomes taut. There's increased tension on those suspensory ligaments. With increased tension on the suspensory ligaments, the lens is pulled into a flatter shape, producing producing a lower refraction, a smaller refraction, a lower conversion, and focusing that light onto the retina. Okay, now this diagram shows that feature again, with relaxed ciliary muscles. Here we can see that there is tension on the suspensory ligaments. The lens is stretched. Okay, so the diagram at the top relates directly to the diagram below. So the ciliary muscles are relaxed, increased tension, and we can see then that the lens is stretched out and flattened. Focusing on a near object with converging rays requires greater refraction. Here we can see that the ciliary muscles contract, they become larger. The suspensory ligaments slacken, so there's less tension on the suspensory ligaments and therefore less tension on the lens, and the lens becomes more spherical, enabling accommodation or a greater degree of convergence. Now, you don't need to know these next couple of slides, but you may be familiar with myopia, short-sightedness, and hyperopia, far-sightedness. So let's have a look at myopia. With myopia, there is too much refraction at the cornea. So a greater refraction at the cornea because the eye is more oval shaped. So there is a greater refraction here. The lens is unable to compensate for that and will continue to refract that light, producing a focal point well before the retina. So there is too much convergence with myopia due to the shape of the cornea. With hyperopia or farsightedness, the eye is much more round. The cornea refracts less. The lens is unable to compensate for that, so will produce a focal point which is beyond the retina. So with distant objects may appear blurred with hyperopia. Near objects may appear blurred with myopia. As I said, you don't need to know this for the exam, but you may be interested. Again, you don't need to know this for the exam, but we can see 3 diagrams here showing normal refraction at the cornea and the lens in the top, producing a focal point on the retina. Nearsightedness, as I said, results in this oval-shaped eye with greater refraction at the cornea, further refraction by the lens, and a focal point which is well before the retina. To treat that, concave lenses can be used, so glasses with concave lenses. Those concave lenses will diverge those rays of light, refract them, causing divergence onto the cornea to a degree that is commensurate, appropriate for the degree of myopia. That compensates for that extra convergence at the cornea, producing diverging rays which focus onto the retina. Farsightedness, like I said, the eye is too spherical. There is too little divergence by the cornea. The focal point is produced beyond the retina. So convex lens. A convex lens would be used in glasses in a person with hyperopia or farsightedness. That causes those rays to converge at the from the lens onto the cornea, so a focal point can be produced on the retina. Okay, if you want to read through this slide, you can do. Just pause the video now, and you can read these. But I think I've explained both of those points. Lastly, laser eye surgery is used to sculpt, to reshape the cornea using the laser to compensate for increased divergence or convergence. Next, we're going to look at the function of the retina and the involvement of the photoreceptor cells. Okay. There are 2 key terms that you need to be familiar with. The first is sensitivity, the second is acuity. Sensitivity is the ability to see at low light intensities, to actually produce an image when there is little light. So high sensitivity means that an image can be produced even when there is very low light intensity. Acuity is similar to resolution, so it means the ability to distinguish detail and to produce a precise and sharp image. Now these 2 terms relate to the 2 types of photoreceptor cell. So the retina contains 2 light-sensitive photoreceptor cells. The rods, which have high visual sensitivity and therefore are able to produce impulses at low light intensities, and the cones, which are involved in colour vision and have high visual acuity. Okay, so this is a summary of the retina and the photoreceptor cells. So the retina contains rods and cones. There are many more rods than there are cones. Those rods are spread across the retina, but are not found in the fovea. So the fovea is the point perpendicular to the pupil and the lens, the point directly behind the pupil. The cones are almost exclusively found within the fovea. Very few are found in the periphery. The rods, there's just one type, so they're unable to distinguish between colours. Cones, there are three types, and we refer to them as a particular colour sensitive cone. So we say that there is a red sensitive cone, a blue sensitive cone, and a green sensitive cone. Rods are sensitive even at low light intensities and therefore have high visual sensitivity. Cones only become stimulated at high light intensities, so have a low visual sensitivity. Rods, and we'll look at why in a moment, have low visual acuity. Cones have high visual acuity. So we looked at a more simplified version of what I'm going to show you next. When light strikes a photoreceptor, either a rod or a cone, that rod or cone cell may be stimulated to produce an impulse. Those impulses travel from the photoreceptor cell, the rod or the cone, to a bipolar neuron. The bipolar neuron then has a synapse, so a second synapse. So there's a synapse between the photoreceptor cell and the bipolar neuron, a second synapse between the bipolar neuron and a ganglion cell. From the ganglion cell, there is another synapse to the sensory neuron, and then from the sensory neuron along the optic nerve to the visual sensory area. So here we can see that. This is part of the diagram that you have. Let me show you on page. Where are we now? Okay, yeah, on page 20. Okay, so we're just going to have a look first at this part of the diagram involving the rods. Okay, so the photoreceptor cells, the rods, have a synapse with a bipolar neuron. You may notice here, though, that each bipolar neuron may be synapsed to more than one rod cell. And this is an important feature of the retina. So the bipolar neuron will form a synapse with more than one rod cell. From that bipolar neuron to the ganglion cell and then the sensory neuron. Okay. This is a very important principle for the function of the retina that you must remember. So many rods synapse to a single bipolar neuron. And if you remember back to the other lesson that you did on the nervous system, you'll know that when a single presynaptic cell forms more than one presynaptic cell forms a synapse with a single postsynaptic cell, we call that spatial summation. Now the advantage of this, and the purpose of it, I suppose, is that it increases the likelihood that the threshold potential will be reached. Meaning that even in low light intensities, it's possible to reach the threshold and to send an impulse from the bipolar neuron via the ganglion cell to the sensory neuron, and therefore an impulse to the visual sensory area in the brain. So the rods, many rods synapse with a single bipolar neuron. That means there is spatial summation. It's more likely that the threshold potential will be reached even in low light intensities, and this accounts partly for the high visual sensitivity. The high visual sensitivity is shown by the rods. Now the cones, again on your diagram on page 20, here we can see that each cone forms a synapse with a single bipolar neuron. There is a 1-to-1 ratio. One cone synapses with one bipolar neuron, and therefore there is no spatial summation. Therefore, at low light intensities, it's less likely that a threshold potential will be reached, and therefore it's less likely that an impulse will be transmitted from the bipolar neuron via the ganglion cell and the sensory neurons to the visual sensory area. Lastly, on your diagram, you have this layer here. This is the choroid. Each of those choroid cells contains lots of melanin, and that melanin prevents internal reflection. So it prevents light from bouncing back across the retina. Now you can do this. You can pause the video here. Looking at your screen, if you focus on the, close one eye, leaving your better eye open. Look at, you probably need to go about 30 centimeters from your screen, perhaps a bit closer. Look at the smiley face, and then from there read along the numbers until the smiley face disappears. Now you may be able to do that, you may not. Sometimes it works, sometimes it doesn't. It takes a little bit of practice. That just demonstrates that at one point of your retina there is a blind spot shown in the graph, I think, which is on, which page of that? Again, page 20 in your notes. So that blind spot is the point at which the retina forms the optic nerve. The blind spot contains no cones and no rods. There are no photoreceptor cells at all within the blind spot. The fovea, also shown on your graph on page 20, shows that the density of rods is 0. There are no rods present in the fovea, but there is a really high density of cone cells. So those cone cells are found almost exclusively within the fovea. And this is really to demonstrate how those two things match up from your diagram you looked at earlier and the graph. Okay, this explanation for visual sensitivity, it's not in your notes, but it might be a really useful one to add to your notes. There is a second feature here about visual sensitivity that I didn't mention before, and that is the pigment, the photosensitive pigment that's found within the rods and the cones. Within rods, the photosensitive pigment is rhodopsin and is can be broken down even at very low light intensities. It's much more unstable and therefore breaks down at low light intensities. The cones contain one of 3 iodopsin pigments. So there is a red sensitive iodopsin, a green sensitive iodopsin, and a blue sensitive iodopsin. All of those require much higher light intensities to break them down, and therefore they contribute to the low visual sensitivity of the cones. So this top row is additional to what I explained earlier on, but it can be useful and is true. Most questions will want comments on these final 3 rows. So the rods, several rods, more than one rod connected to a single bipolar neuron. Light may stimulate several of those rods, causing spatial summation at the bipolar neuron. Therefore, threshold is more likely to be reached and impulses more likely to be sent to the brain. That accounts for the high visual sensitivity in rods. Cones, on the other hand, have low visual sensitivity. This can be explained by each cone forming a synapse with a single bipolar neuron. There's no spatial summation, as we've got this 1-to-1 ratio. Therefore, threshold is less likely to be reached at low light intensities, and therefore it's less likely that impulses will be received at the visual sensory area of the brain. Pause the video now if you want to copy this down. Okay, this next diagram begins to explain why cones have a much higher visual acuity. Now it's related again to those synapses between the photoreceptor cells, the cones on the left and the rods on the right, and the bipolar neurons. Each cone has a 1-to-1 ratio with a bipolar neuron. So if 2 separate images fall on the cones shown here and here, providing those images have a high enough light intensity to produce an impulse, that will result in 2 separate impulses travelling along the sensory neuron to the brain, and the brain will then interpret those 2 images as separate. Whereas in rods, where we have that convergence, so spatial summation, many rods forming a synapse with a single bipolar neuron, those 2 separate images may fall on 2 rods. However, both of those rods being synapsed to a single bipolar neuron means that only one impulse will be received at the visual sensory area, and those 2 separate impulses will be perceived as one. So, cones can distinguish between 2 close points of light, whereas rods cannot. So here's a summary of that for visual acuity. In rods, several rods connected to a single bipolar neuron. Convergence occurs. That's when impulses from many converge into one bipolar neuron. Therefore, light stimulating rods will result in only one set of impulses being sent to the brain. Okay, all of that contributes to a low visual acuity by the rods. On the other hand, the cones, each cone is connected to one bipolar neuron. There will be no what's referred to as retinal convergence. The light stimulating each of those cones results in separate impulses being sent to the brain, and the visual sensory area and then the visual association area will perceive those as separate images. Okay, next. Is it possible to see colour in the dark? No, is the straightforward answer, I suppose. Cones are responsible for colour. In the dark, cone cells will not be stimulated because they have very low visual sensitivity. In the dark, only the rod cells will be stimulated, but because there is only one type of rod cell and one type of rhodopsin, those rod cells are not responsible for perceiving or producing impulses to perceive colour. So it is not possible to see colour in the dark unless that colour is from a bright light intensity. So we're going to move on now to colour vision. And this involves three types of cone. The red, sensitive, blue, sensitive, and green, sensitive cones. Now I'm really going to emphasise this point. We cannot refer to them as red cones, blue cones, and green cones. You must refer to them as red, blue, or green sensitive cones. Each one of those types of cone cell contain one of the three different iodopsin pigments. So red sensitive cones, understandably, contain red sensitive iodopsin. Blue sensitive contain blue sensitive iodopsin. Green sensitive contain green sensitive iodopsin. Okay, so there is only one type of iodopsin within each type of cone cell. Each pigment has its own absorption peak that corresponds to these three wavelengths of light. I.e., there is a wavelengths of light which are red, wavelengths of light that are blue, and wavelengths of light that are green, and they will stimulate one of the types of cones or one of the types of iodopsin. Okay, so this graph you can find on, where is it? There we go, on page 25. Okay, and it demonstrates what's called the trichromatic theory. On the x-axis, we have wavelengths of light. Each wavelength of light will correspond to a different colour. The shortest wavelengths of light are those colours at the blue end of the visible spectrum. Longer wavelengths of light are at the orange and red, or at the red end of the visible light spectrum. That's on the x-axis. On the y-axis, it shows how sensitive each type of cone is to a particular type of to a particular wavelength, and that is measured as the amount of light absorbed as a percentage of the maximum. So blue sensitive cones will absorb 100% of wavelength of light around 430 nanometers. Okay, where's my pen not working? Okay, so about this stage, the absorption, this is called the absorption peak. The absorption peak for those blue sensitive cones is about 430 nanometers. Green sensitive cones, it's higher than that, so it's around perhaps 550 to 570 nanometers. And then the red sensitive cones, around 590 up to perhaps 600 nanometers. So, different wavelengths of light may result in a different degree of stimulation for each type of cone. Impulses are then sent to the visual sensory cortex in the brain, where the relative stimulation of each type of cone is processed to produce the range of colours which we perceive. We've got this little nota bene down the bottom here. Colour itself does not exist, just different wavelengths of light, and those wavelengths of light will be perceived at the visual association areas. So you also have this table in your notes, and it shows how each colour can be perceived. So white light is a mixture of all of the colours of the light spectrum, visible light spectrum, and therefore they contain blue light, green light and red light, or wavelengths which are blue, green and red. White light will stimulate the blue sensitive cones, the green sensitive cones and the red sensitive cones equally to produce what we perceive as white. Black, at the other end of the table, is the absence of light. So the blue sensitive, green sensitive and red sensitive cones are not stimulated, and therefore no colour is produced, and that is perceived as black. Everything in between is a result of the degree of stimulation of each type of cone. So blue light will only stimulate blue sensitive cones, and therefore the colour perceived will be blue. Likewise, green only stimulates green sensitive cones and is perceived as green. Red light, or wavelengths, which are red, stimulate the red sensitive cone and will only be perceived as red. Any other colours are combinations of stimulation, and it's the relative amount of stimulation of each of the different types of cone which will determine which colour is perceived. So if blue sensitive and red sensitive cones are stimulated, the colour perceived will be purple. If there is green sensitive, perhaps more green sensitive stimulation, let's put an extra tick here, with some red sensitive stimulation, the colour perceived would be yellow. If there is more red sensitive but some green sensitive stimulation, the colour perceived will be orange. Okay, now in your notes, let me just go back. You have this sentence here. Impulse is sent to the visual sensory cortex, where the relative stimulation of each type of cone is processed to produce the range of colours which we perceive. That means how much colour is, or how much of each type of those cones is stimulated will determine which colour is perceived. Okay, now the next couple of slides I'm going to show you are about colour blindness. And now again, you don't need to know this, but this is often an interesting topic for our students. Okay, so colour blindness results in the inability to perceive certain colours. It may be due to a lack of a particular type of cone cell, red sensitive or blue sensitive or green sensitive, cones which lack the particular iodopsin pigment, or iodopsins which are different and therefore absorb a different wavelength of light. Iodopsin is part protein, the opsin part is a protein, and part retinal, which is a derivative of vitamin A. If that protein has a slightly different tertiary structure, it may absorb a slightly different wavelength of light. Now, if those wavelengths of light overlap with others, it may be difficult to distinguish. So that can almost be linked to this fourth point. The absorption peaks may overlap too much, so it's difficult to distinguish between the two colours. Okay, here we have a similar graph to the one we saw earlier on. If there is blue light, say 420 nanometers, only the blue sensitive cones will be stimulated. Here we go. If there is light which is a combination of green and red, that may produce a colour, so 500 nanometers, we get a stimulation of green and red, may produce a cyan colour. Orange is a result of those green and red cones being stimulated again, but with greater red stimulation here. I think actually this should probably be more like yellow rather than cyan there, but never mind. Okay, here we have different types of colour blindness. Normal colour vision at the top, deuteranopia, protanopia and tritanopia. Non-colourblind person would have those absorption peaks that we looked at earlier on. Someone who is with protanopia, we can see here is missing that red sensitive pigment, and therefore will be red colourblind to red. So that is full protanopia. There is protananomalous, which means that they may still have that red photosensitive pigment, but it's too similar to the green and therefore is not distinguishable. Deuteranopia, okay, here we're missing the green sensitive pigment, so unable to see green. Deuteranomalous colour vision means when the green sensitive pigment is modified and it's difficult to distinguish from red. Last thing to do here, can you please stare at this? If you pause the video now, in fact no, don't pause. Stare at this for 30 seconds. Now you can either do that by pausing it, ignore what I said about pausing it, just watch the red spot. Okay, keep staring at the screen. And then what colour do you perceive on the white background there? So what did you see? Hopefully you should have seen a blue-green or a cyan circle on the white screen. Now can you explain why? Well, staring at the red cycle causes the red sensitive receptors to become bleached. So all of the red sensitive iodopsin in the red sensitive cones is broken down. When the screen turns to white, only the blue and the green sensitive receptors can be stimulated. The screen will only appear white again once the iodopsin in the red sensitive cone has been reformed. And that's that. Okay, so that's everything on the eye. If you have any questions, you can always email me, and I will speak to you soon. Okay, take care. Bye-bye.